Dear students, welcome to the massive open online course on human genetics. Today we will be talking about the introduction to heredity and genetics. The main objectives of this module are to know about the brief history of genetics, to know about genotype and phenotype, to know about the genetic variation and heredity in organisms, to know about the inheritance patterns in genetics and use of Punnett squares, to know about the causes and effect relationship of gene transmission from parents to offsprings. As we know, all organisms have genetic material in the form of DNA or RNA that passes from one generation to the next generation. During this process, the genetic material of the organism undergoes change. The rate and kind of change depend on the environment, and this brings the variation in organisms. They enable the organism to adapt themselves in changing environment. An accumulation of variations in the individuals is because of heredity, and it leads to the evolution of species by natural selection. The genotype is the set of genes that an offspring inherits from both parents, which is also called as the genetic material of an organism. Whereas phenotype is the external appearance of an organism. It depends on the complex interactions between genes and their environment that leads to genetic variations, which in turn makes the phenotypic variations. Any variation in the DNA of the germ cells can be inherited. Even though 99.6% of the DNA is same in all individuals, what makes change in the individual appearance and personality is the variation. Favorable characters are transmitted from one generation to another, leading to evolution. The interaction between genetic variation in a population and the environment results in natural selection. Gene flow also helps in genetic variation by introducing new genes into a population. Genetic variations can also result due to sexual reproduction. This occurs during recombination of in homologous chromosomes during meiosis process of cell division. Variations forms the basis of heredity, and they form raw material for evolution and development of new species. So variation in the phenotype is required for the process of evolution by natural selection. So natural selection affects the genetic architecture of a population indirectly through the contribution of phenotypes. Genotype along with the environment gives the phenotype. The phenotype varies according to the environment. Now let's discuss the background of modern genetics first. The modern genetics started with the work of the Austrian monk Gregor John Mendel. His work on pea plants, published in 1866, described what came to be known as Mendelian inheritance. In the centuries before and for several decades after Mendel's work, a wide variety of theories of heredity proliferated and marketed in the 1900s. That was the first among them was the rediscovery of Mendel independently by three scientists, Hugo de Vries, Karl Correns, and Erich von Teschermark in 1915, marked the basic principles of Mendelian genetics, and that had been applied to a wide variety of organisms. Most notably among them is the fruit fly or the Drosophila melanogaster. Led by Thomas Hunt Morgan and his fellow Drosophilists, geneticists developed the Mendelian model, which was widely accepted by 1925. Alongside experimental work, mathematicians developed the statistical framework of population genetics, bringing genetic explanations into the study of evolution. With the basic patterns of genetic inheritance established, many biologists turned to investigations of the physical nature of the gene. In the 1940s and early 50s, experiments pointed to DNA as the portion of chromosomes and perhaps other nucleoproteins that held genes. A focus on new model organisms such as viruses and bacteria, alongside the discovery of the double helical structure of DNA in 1953 by Watson and Crick, marked the transition to the era of molecular genetics. In the following years, chemists developed techniques for sequencing both nucleic acids and proteins, while others worked out the relationship between the two forms of biological molecules, the genetic code. The regulation of gene expression became a central issue in 1960s. By the 1970s, gene expression could be controlled and manipulated through genetic engineering. In the last decades of the 20th century, many biologists focused on large-scale genetics projects and sequencing entire genomes, the example of which is the Human Genome Project, followed by many others, or the HapMap project. The concept of heredity and the ability to pass on traits from one generation to next is central to genetics as well as to evolution. This activity will cover inheritable traits, dominant and recessive traits, genotypes and phenotypes, and Punnett squares. In nature, there are two basic methods of passing genetic information from one generation to next generation. One is that sexual reproduction, and the another one is asexual reproduction. In asexual reproduction, one parent makes an exact copy or a clone of itself. This is commonly done through splitting one cell in half, that is binary fission or fragmentation. Asexual reproduction is used by bacteria, many plants and fungi, and some animals, generally simpler organisms. Some organisms can reproduce either sexually or asexually depending on condition of their life history. That is the phases. In some cases, more advanced organisms such as sharks have shown the ability to reproduce asexually through a process called parthenogenesis. Sexual reproduction, on the other hand, involves the combining the genetic material from two parents, that is the male and the female, or the mother and the father. So sexual reproduction increases the variability in the gene pool by combining the DNA from these two different organisms. This allows important information to be passed down through many generations. An offspring will get half of its DNA from its mother and half of its DNA from its father. The offspring's mother got half of her DNA from her mother and half from her father. This pattern goes back through countless generations. Because the DNA is combined in many new ways by having DNA from two individuals combining to form the offspring, new characteristics can be formed. This is significant because this variability will make the species more able to survive if the environment changes. All aspects of an organism are controlled by its DNA. So this includes things that we can see and things that we cannot. When thinking about heredity, it is often helpful to focus on the things that we can see, that the traits are characteristics of an organism that have natural variety. So this doesn't really apply to things like how cells divide, because this is similar among all the cells. Additionally, traits generally refer to things that we can see, that is eye color, hair color, skin color, attached earlobes, tongue rolling, etc. So each version of a trait is called an allele. For example, eye color is a trait. The alleles for eye color are brown eyes and blue eyes. Each offspring gets one set of alleles for each trait from its mother and one from its father. So each parent has two sets of alleles, one from each parent, that it could potentially pass on to his or her offspring, although each parent will only pass on one allele for each trait. This is the basis of sexual reproduction and variability. Every organism that is a result of sexual reproduction has two copies of every gene. So each organism has two copies of each gene, one from the mother and one from the father, that it can pass along from to its offsprings, which will be combined with the DNA from another organism when they reproduce. As a parent, only one copy of each allele that a parent possesses will be passed on to each offspring. This is why siblings are often similar but not identical. So each organism that is the result of sexual reproduction has two copies of each allele. The interaction of these alleles will determine the traits that we see. The alleles can be same, that is homozygous, for a given trait, or different, that is heterozygous, for a given trait. The way that alleles interact is referred to as the dominance pattern for a given trait or character. In humans, the alleles for brown eyes is dominant to the allele for blue eyes. So brown here is dominant to blonde here. In Gregor Mendel's early experiments of genetics, purple flowers on pea plants were dominant to white flowers. The dominant allele is the one that is expressed, that is what we can see or visualize. It is possible for someone to be a carrier of a recessive allele but not show the trait. This is how two parents with brown eyes can have a child with blue eyes. This would occur if someone is heterozygous for a given trait, one allele for blue eyes and one allele for brown eyes. Only the dominant allele, that is with brown eyes, will be expressed, and the recessive allele, that is with blue eyes, will be hidden. However, because the individual carries the recessive allele, and in sexually reproducing organisms, the parent randomly passes on allele to each offspring, it is possible for offspring to show versions of traits not seen in the parents. This difference between what your genes say and what you look like are referred to as your genotype and your phenotype. So genotype is what your genes or alleles actually say, and the genotype always includes two alleles. The genotype can be homozygous dominant, that is two alleles for the brown eyes, or homozygous recessive, two alleles for the blue eyes, or heterozygous, that is one allele for brown eyes and one allele for the blue eyes. However, the phenotype is what you look like. It only includes the version of the trait or the character that you see, either blue or brown. The dominance patterns aren't always completely straightforward. Incomplete dominance occurs when two alleles combine to form a phenotype that is different from each of the parents. A well-known example of this is in snapdragons. White and red flowers both exist, but when an individual has a white allele and a red allele, the phenotype is pink flowers. In genetics, we use Punnett squares to determine the possible genotypes and phenotypes that can result from a given cross of a parent. It allows us to determine the probability that certain parents will have offsprings with a certain trait. This is important when thinking about things like genetic diseases if a couple is considering of having children. Dear students, let's now discuss about Punnett squares. Punnett squares work by placing each possible allele associated with each box. So the top represents the two alleles that one parent can pass on to offsprings, and the left side that represents the two alleles that the other parent can pass on. The boxes are filled with the possible allele combinations. In general, this is done using letters to represent a given trait. Capital letters represent dominant traits and lowercase letters represent the recessive traits. Only one letter is used even if the allele don't start with the same letter. For example, if you are looking at white flowers and purple flowers, one could use P to represent purple alleles and P to represent white flowers, even though there is no P in the white. In the case of incomplete dominance, one letter represents the trait and superscripts represent each possible allele. An example of this scheme is seen in the gummy bear genetics activity. Dear students, let us now discuss the inheritance patterns. Inheritance patterns fall into three general categories. One is autosomal inheritance, based on the variations of a single gene or chromosome. Next is the sex-linked inheritance, based on the variation of a single gene or sex chromosome. The third one is non-Mendelian inheritance, which is based on the variation of a single gene or organelle chromosomes. The basic laws of inheritance help us to appreciate how traits are passed on from one generation to the next generation. Here, the aspects going to be emphasized are: one is autosomal dominant inheritance. When the dominant allele is located on one of the 22 pairs of autosomes, non-sex chromosomes, we refer to its inheritance pattern as autosomal dominant. An example of an autosomal dominant disorder is neurofibromatosis type 1, a disease that induces tumor formation within the nervous system that leads to skin and skeletal deformities. Consider a couple in which one parent is heterozygous for this disorder and who therefore has neurofibromatosis, that is NN, capital and small, and one parent is homozygous for the normal gene, that is small nn. The heterozygous parent would have a 50% chance of passing the dominant allele to this disorder to his or her offspring, and the homozygous parent would always pass the normal allele. Therefore, 4 possible offspring genotypes are likely to occur equally, that is capital N, small n, small nn, and small nn. That is, every child of this couple would have a 50% chance of inheriting neurofibromatosis disease. This inheritance pattern in a child from the Punnett square is named after its creator, the British geneticist Renegade Punnett. So now the autosomal recessive inheritance. When a genetic disorder is inherited in an autosomal recessive pattern, the disorder corresponds to the recessive phenotype. So heterozygous individuals will not display symptoms of this disorder because their unaffected gene will compensate. Such an individual is called a carrier, that is, carriers for an autosomal recessive disorder may never know their genotype unless they have a child with the disorder. An example of an autosomal recessive disorder is cystic fibrosis. Cystic fibrosis is characterized by the chronic accumulation of a thick, tenacious mucus in the lungs and digestive tract. Decades ago, children with cystic fibrosis rarely lived to adulthood. With advances in medical technology, the average lifespan in developed countries has increased into middle adulthood. Cystic fibrosis is a relatively common disorder that occurs in approximately 1 in 2,000 Caucasians. A child born to 2 cystic fibrosis carriers would have a 25% chance of inheriting the disease. This is the same ratio that is the dominant versus recessive, that is 3:1, that Mendel would have observed in his pea plants. The pattern is shown in the diagram that tracks likely incidence of an autosomal recessive disorder on the basis of parental genotypes. On the other hand, a child born to a cystic fibrosis carrier and someone with two unaffected alleles would have a 0% probability of inheriting cystic fibrosis but would have a 50% chance of being a carrier. Other examples of autosomal recessive genetic illnesses or disorders include the blood disorder, that is sickle cell anemia, the fatal neurological disorder Tay-Sachs disease, and the metabolic disorder phenylketonuria. Now let us discuss the X-linked patterns of inheritance. An X-linked transmission pattern involves genes located on the X chromosome of the 23rd pair. Recall that a male has one X and one Y chromosome. When a father transmits a Y chromosome, the child is male. When he transmits an X chromosome, the child is female. A mother can transmit only an X chromosome as both her sex chromosomes are X chromosomes. Now let us discuss the X-linked dominant inheritance. When an abnormal allele for a gene that occurs on the X chromosome is dominant over the normal allele, the pattern is described as X-linked dominant. This is the case with vitamin D resistant rickets. An affected father would pass the disease gene to all of his daughters but none of his sons because he donates only the Y chromosome to his sons. If it is the mother who is affected with this disorder, all of her children, that is the male or female, would have a 50% chance of inheriting the disorder because she can only pass an X chromosome on to her children. For an affected female, the inheritance pattern would be identical to that of an autosomal dominant inheritance pattern in which one parent is heterozygous and the other is homozygous for normal gene. Now let us discuss the X-linked recessive inheritance. X-linked recessive inheritance is much more common because females can be carriers of the diseases yet still have a normal phenotype. Diseases transmitted by X-linked recessive inheritance includes color blindness, the blood clotting disorder hemophilia, and some forms of muscular dystrophy. For example, X-linked recessive inheritance, consider parents in which the mother is an unaffected carrier and the father is normal. None of the daughters would have the disease because they receive a normal gene from their father. However, they have a 50% chance of receiving the disease gene from their mother and becoming a carrier. In contrast, 50% of the sons would be affected. With X-linked recessive disease, males either have the disease or are genotypically normal, they cannot be carriers. Females, however, can be genotypically normal, a carrier who is phenotypically normal or affected with the disease. A daughter can inherit the gene for an X-linked recessive illness when her mother is a carrier or affected or her father is affected. The daughter will be affected by the disease only if she inherits an X-linked recessive gene from both the parents. As you can imagine, X-linked recessive disorders affect many more males than females. For example, color blindness affects at least only 1 in 20 males, but about 1 in 400 females. Dear students, let us now discuss the non-Mendelian inheritance. Non-Mendelian inheritance is any pattern of inheritance in which traits do not segregate in accordance with the Mendel's laws. These laws describe the inheritance of traits linked to a single gene or chromosome in the nucleus. In Mendelian inheritance, each parent contributes one of two possible alleles for a particular trait. If the genotypes of both parents in a genetic cross are known, Mendel's laws can be used to determine the distribution of phenotypes expected for the population of offsprings. There are several situations in which the proportions of phenotypes observed in the progeny do not match with the predicted values. So the non-Mendelian inheritance plays a role in several diseases which affected the processes, like incomplete dominance. In incomplete dominance, the traits blend together, producing an intermediate phenotype. If two flowers are crossed together, a hybrid will be produced that is in between both the parents. For example, in snapdragon plant, if a homozygous white flower is crossed with a homozygous red flower, a pink flower is obtained. Dear students, let's now discuss the codominance. When the two alleles present are simultaneously expressed, it's known as codominance. For example, in some variety of chicken, the alleles for black feathers are codominant with alleles for white feathers. If a black chicken is crossed with a white chicken, a chicken with both white and black feathers are obtained. Now, let's discuss the types of codominance. The one example is multiple alleles. Some populations have multiple alleles of a given gene. For example, the gene for coat color in rabbits has 4 common alleles: C, CCH, CH, and C. Dear students, let's now discuss the pleiotropy. When one gene affects multiple characteristics, it is known as pleiotropy. For example, Marfan syndrome results in several symptoms such as very tall height, thin fingers, heart problems, dislocation of limbs, etc. These symptoms are not related directly but are caused by the mutation of a single gene. The next one is lethal alleles. A few genes have alleles that affect the survival of the organisms. For example, lethal yellow occurs due to a mutation in mice that turns its coat yellow. Mice with homozygous alleles die during embryonic development. So lethal alleles can be dominant or recessive and can be expressed in homozygous or heterozygous conditions. Dear students, let's now discuss the polygenic inheritance. There are a few traits that are controlled by many genes. For example, our height is controlled by more than 400 genes. Skin pigmentation is the result of several genes. Dear students, let's now discuss the extranuclear inheritance. It is the transmission of genes that occurs outside the nucleus. That is, the mitochondria DNA is passed on from mother to offsprings. It usually occurs in cytoplasmic organelles such as mitochondria and chloroplast. It's a form of non-Mendelian inheritance discovered by Carl Correns. The mitochondria disease are also passed on to the offsprings. This does not follow Mendel's laws. Dear students, let's now discuss the scope of genetics and heredity. As soon as fundamental principles of genetic inheritance were clearly established in the early 20th century, anthropologists began using these principles and new empirical data to illuminate long-standing problems of human variation and primate phylogeny, initially focusing on human blood characteristics. Geneticists quantified regional differences in ABO genotype and tried to correlate these differences with more traditional osteometric assessments of variation. Many correlations have been found between pathogenic diseases and genetic variations. Different patterns of genetic variation among populations are explained as accidents of demographic history or founder effects rather than as adaptive responses of the gene pool. More generally, understanding the role that genetic variations may play in healthcare involves the analysis of risk factors and consequent probabilities. The science of heredity or genetics is the study of two inconsistent elements of nature, that is, the heredity and variations. We study the preserved genes and compare the genome of different species to uncover similarities and differences that improve their understanding of how human genes function and are controlled. This knowledge helps researchers develop new strategies to treat and prevent human diseases. Dear students, with this, we come to the end of this module on Introduction to Heredity and Genetics. Hope you have understood it well. See you with another module. Till then, bye bye.